Bismillahirrahmanirrahim. Assalamu alaikum class. The topic of the today lecture is the antibiotics. This topic was already started in the regular classes, which was discontinued due to the disturbance. Now I am just briefly giving the point or recalling those points which we have already covered. An antibiotic is a substance which is obtained from one living organisms, from one living system, and are used to inhibit the growth or to kill another type of the microorganisms are used against another living system in a very minute quantity. Before the discovery of the antibiotics, and few chemicals were used for this purpose. For example, we have got the antibacterial sulfonamides. In 1987, Pasteur has reported few compounds from the mold, but due to higher toxicity, those compounds have no importance clinically. In 1928, Fleming accidentally discovered the antibacterial role of Penicillium notatum. In 1938, this was isolated by freeze-drying method. The name of the scientist was Fleury and Chain. Scientists were Fleury and Chain. From 1944, penicillin was used clinically, and in 1945, structural elucidation was carried out by X-ray diffraction technique. The first reported penicillin was in 1957. Between the 58 and 60, a biosynthetic intermediate, that is 6-APA, was obtained, which is also an intermediary compound in the synthetic or biosynthetic pathway by the Sheehan. Sheehan was the scientist who has reported the first synthesis in 1957. Now 6-APA or 6-aminopenicillic acid is obtained by fermentation, or they are obtained by hydrolysis of penicillin G or penicillin V, and after acylation of this 6-APA, we are going to have got different derivatives. The first compound of the penicillin which was obtained was the penicillin G. That is also termed as penicillin G or benzyl penicillin. And then we have got the penicillin V or phenoxymethyl penicillin, phenoxymethyl penicillin, these two penicillins were obtained by the addition of different carboxylic acids in the corn steep liquor. In the case of penicillin G, phenyl acetic acid was added in the fermenter, while in the case of penicillin V, phenoxyacetic acid was added in the medium, and we have got phenoxymethyl penicillin. These two penicillins, penicillin G and penicillin V, are termed as natural penicillins. The penicillins are also termed as beta-lactam antibiotics, as they have got a four-membered lactam ring. Sometimes they are termed as PNAMs, as they have got an unsubstituted 4 and 5 membered fused rings. The numbering is also an interesting one. Generally, we are using the numbering according to chemical abstract, in which the priority is given to the sulfur, or designated as number 1. In the USP, nitrogen is designated as 1 and sulfur as 4. Sulfur as 4. The penicillins have got the beta-lactam ring, or 4-membered cyclic amide, which is fused with a 5-membered thiazolidine ring, which is fused with a 5-membered thiazolidine ring. Thiazolidine ring is substituted at 6th position by 2 methyl groups, while at 3rd position carboxylic group is there. The portion having 2, 3 and 4 parts, or the part having the dimethyl group, carboxylic group and the nitrogen, is obtained from the valine, while the other portion is obtained by the amino acid cysteine. The penicillin G have got a number of problems. Main 4 major problems were identified. It is acid sensitive. What does it mean? Means it cannot be taken orally. It is going to be hydrolyzed by gastric acid. The second problem is the lactamase sensitivity. Beta-lactamase degraded the beta-lactam ring, or opening or cleavage of the ring is there. The third problem is the narrow spectrum of the penicillin G. The penicillin G is active against gram-positive organisms, almost not active against any gram-negative organisms. The fourth problem is the allergenicity of these penicillins, and still this problem is there. Fortunately or unfortunately, but fortunately the other three problems are resolved. If we are going to take the different penicillins, for example the benzyl penicillin, in which penicillin G, in which the benzyl group is substituted in the acyl amino side chain, the compound is acid sensitive. It is also beta lactamase sensitive. It is narrow spectrum and also have got the allergenicity. To overcome these problems, we are going to modify the structure of the penicillin G. But if we studied the SAR of penicillin, we have found that in the penicillin, the fused ring system is essential for the activity. Beta-lactam ring separately is also essential. Free carboxylic group is essential. In few derivatives, carboxylic group is changed, but this type of the compound served as a prodrug. What does it mean? Means after the metabolism or by means of metabolism, the carboxylic group is going to be generated, or for the active molecule, free carboxylic group is essential. The acyl amido side chain is also essential. We have got one of the example of thianamycin. The acyl amido side chain is not present. Sulfur is required, but sulfur is not essential. The amino group at the carbon 6 is required for the activity. Substitution on acyl group will increase or will provide the more potent compounds. Stereochemistry is also important. The beta and the alpha position, the beta and alpha groups is important. In the same way, I say that very few positions are available for the substitution, or very little variation is tolerated by the main nucleus of penicillin. To overcome the first problem, we have come to know that the beta lactam ring, which is essential, is going to be cleaved by the action of gastric acid, but this degradation is also observed in the case of penicillin G by itself. What does it mean? In penicillin G, we can observe the self degradation mechanism. The carbonyl group in the acyl amino side chain serve as a nucleophile and attack the carbonyl group of the beta lactam ring. We have come to know that a simple strategy can be used by the substitution of an electron withdrawing group such as chloro, bromo, nitro, and heterocyclic structure. We can overcome the problem. These groups will withdraw the electrons from the carbonyl of the acyl amino side chain, and hence this molecule not serve as a nucleophile for the carbonyl group of the beta lactam ring, or the neighboring attack is prevented, or the carbonyl group of the acyl side chain will not behave as a nucleophile, not attacking the carbonyl group of the, and hence the acid sensitivity can be overcome. By using this strategy, the compound which we have got is the penicillin V, in which we have got the phenoxymethyl group, which serve as an electron withdrawing group, and hence the penicillin V is not acid sensitive, but still it is lactamase sensitivity. It is lactamase sensitive. It has got a narrow spectrum activity, and also allergic reactions are there, but can be taken orally. In 1960s, that was known as a very crucial or critical year in which more than 80% of the infections by the Salmonella aureus in the hospitals are not treated or were not treated because they have got the resistance against the available antibiotics or the compounds. To overcome this beta lactamase sensitivity, we have addressed the problems, and it was resolved that by addition, by substitution of a bulky group, a group which physically hinder, prevent the access of the beta lactamases to the target or the carbonyl group of the beta lactam ring. Hence, the bulky groups provide the shielding effect, the steric hindrance, and the cleavage, the opening, the breaking was or is prevented, and hence we have overcome the problem. Methicillin was the first compound used clinically which have got the bulky group, that is dimethoxyphenyl group. Dimethoxyphenyl group that will serve as a bulky group, provide shielding effect to the carbonyl group of the beta lactam ring. Methicillin have got the resistance or provide the protection against beta lactamases, but the compound is not resistant to acid, or still it is acid sensitive and cannot be taken orally. In the same way, narrow spectrum and allergic reactions are there. In this series, we have got the augacillin, which have got I at R if we consider the structures which we have already provided. In the cloxacillin, one R is Cl and the other R or R dash is H. In dicloxacillin, R and R dash are Cls. In flucloxacillin, R is Cl, R dash and F. Then in the next series, that is termed as second generation of the penicillins, a hydrophilic group was substituted in the acyl amidoside chain. How we have got that lead, how we have used that molecule, hydrophilic molecule. This was identified by observing the behavior of membranes, cell membranes of gram-negative and gram-positive organisms. We have observed that the hydrophilic groups can easily penetrate, can easily penetrate through the cell wall of beta gram-negative organisms. can enter easily through the gram-negative cell membrane. The first reported compound was the ampicillin, that is also termed as aminopenicillin, the first molecule in the second generation of penicillins. In this molecule, we have got the amino group, which serve as a hydrophilic group, and also this amino group have got the electron withdrawing effect. Hence, it is active against gram-negative organisms. It is also resistant to gastric acid, but not have got any bulky group, hence not resistant to beta lactamases, not resistant to beta lactamerases. In the previous examples, we have got the derivatives of oxacillin, in which the modification in the molecule have changed the rate of the absorption, as cloxacillin is better absorptive rate. The same way, flucloxacillin have got less protein binding affinity. In the series of